Hi, this is Sudarshan here. I've recently joined in-STEM at the Center for Inflammation and Tissue Homeostasis. A bit about me: I was born and brought up in Bangalore, and I did my PhD from the Indian Institute of Science here in Bangalore, after which I moved to Institut Curie in Paris for my postdoctoral studies. Now, I stayed in Paris for about 8 years before moving back to Bangalore to join in-STEM. I have always been interested in understanding the functions of mammalian cilia and flagella, the tiny hair-like structures that are found on most epithelial cells in mammals. Now, I learned about cilia and flagella during my graduation, and I was intrigued to know more about them as to what regulates their functions and their structure and so on. This is what I did when I joined my postdoctoral lab, which was focusing on understanding how tubulin post-translational modifications impact the functions of cilia and flagella. Now, tubulin forms the microtubules which form the core structures of cilia and flagella, and I focused on a particular modification called glycylation, which is the addition of glycine amino acids onto the microtubules, because glycylation is one particular modification that happens exclusively on cilia and flagella. So I wanted to know more about how this impacts the functions. There is a lot of research that's going on that shows that defects in cilia can lead to various human disorders. Now, in the lab, I'm focusing on a new aspect of it as to how microtubules and their post-translational modifications are impacting the functions of cilia. In a mouse model where we removed the enzymes responsible for this modification, we found that the male mice were subfertile. And when we went ahead to look at the cellular and the molecular aspects of this, we realized that the sperm had difficulty in motility, and they were actually swimming in circles compared to the wild-type sperm that swim in a straight line. Now, this was fascinating for us because for the first time, we were able to establish a molecular role for glycylation in mammals to say that they impact the function of the sperm. Now, I am interested to know more about how not just glycylation but other tubulin modifications also impact the functions of cilia and flagella in mammals. Now, homeostasis, on the broader aspect, is all about how the tissue is able to regulate its function and how it's able to regenerate or repair itself post-injury or infection. Now, this is what the groups within the theme are looking at already. And what is also known is that many of the cells that are involved in these processes are all ciliated. So by trying to understand the role of cilia in this whole process of homeostasis and how tubulin modifications impact the functions of cilia in this process will give a new perspective to the understanding of tissue homeostasis as a whole. Now, in the long term, what I would want to address is to understand the molecular aspects regulating the homeostasis through the cilia to be able to develop new therapeutic strategies to combat diseases like retinal degeneration and kidney dysfunction. Now, as I mentioned earlier, cilia and flagella are found on almost all organs of the human body. So by making people understand this aspect of my research, I will also be able to emphasize that understanding the role of tubulin modifications is equally important to see if defects in these modifications can lead to the kinds of disorders, be it the respiratory dysfunction, retinal degeneration, kidney dysfunction, and so on. So come along, visit us in the lab, see what we do. See you around.